Okay, welcome to chapter 15 on translation. We're moving through the central dogma at this point, and we want to start out with a discussion of a condition known as asplenia. Right? The spleen's an organ found in the upper abdomen on the left-hand side of the body under a muscle called the diaphragm. And isolated congenital asplenia is an autosomal dominant condition where children are born without a spleen. A small group of children are born without spleens, and these kids are highly susceptible to life threatening bacterial infections, and many die in childhood. This rare disorder, known as isolated congenital asplemia, or ICA, is inherited as an autosomal dominant. Except for the absence of a spleen, children with ICA are unaffected, but their immune function is severely compromised. When infected with bacteria that the immune system normally eliminates, these children develop raging infections that quickly spread throughout the body. Even when treated with modern antibiotics, they often die. The reason for this is that one of the roles of the spleen is to remove pathogens from the blood directly. Okay, so there's always going to be a percentage of pathogens that make it through the skin and the mucous membranes and into the blood, and it's those that are normally siphoned out by the spleen. Using DNA sequencing, the coding DNA of 23 individuals with ICA was compared to DNA from normal spleens. Statistical analysis pointed to differences in one particular gene associated with ICA encoding a ribosomal protein, RPSA. This protein is one of the 33 proteins that make up the small subunit of the ribosome, which is the organelle responsible for protein synthesis. How a defect in the RPSA gene results in the absence of a spleen isn't known, but diseases such as ICA, which result from defective ribosomes, are referred to as ribosomopathies. Researchers found that several different types of mutations in RPSA associated with ICA. Some cause premature stop codons, halting translation before a functional protein could be made. One was a frameshift mutation, a change that alters the way the messenger RNA sequence is read during translation, and others change the amino acid sequence of the RPSA protein itself. One interesting but unanswered question is why a defect in the RPSA affects only the spleen. Inherited mutations in RPSA occur in every cell of the body, and protein synthesis, which is carried out by ribosomes, is essential for numerous life processes, yet these mutations affect only the development of the spleen. Why aren't other organs changed? Why aren't numerous physiological functions affected? We still don't know the answers to these questions. Beadle and Tatum used Neurospora to study biochemical results of mutations. Neurospora is easy to cultivate in the lab, and the main vegetative part of the fungus is haploid, which allows the effects of recessive mutations to easily be observed. Wild-type Neurospora grows on minimal medium, which contains only inorganic salts, nitrogen, and a carbon source, like sucrose, and the vitamin biotin. The fungus can make all the biological molecules that it needs from these basic compounds, but mutations may arise that disrupt fungal growth by destroying the fungus's ability to make one or more essential biological molecules. These nutritionally deficient mutants, which are called auxotrophs, can't grow on minimal media, so they can grow on media that contains a substance that they no longer are able to produce. Okay, so this is one of the ways that we can determine whether or not a biosynthetic pathway is fully functional. Okay, is by having media where we can define exactly what's in it. Okay, and if the organism is incapable of making what's missing in the media, then it won't grow, whereas it would grow on complete media. Okay? Beadle and Tatum irradiated the spores of Neurospora to induce mutation and then placed the spores in different culture tubes with complete media, which contains all the biological substances needed for growth. These spores grew into fungi and produced spores by mitosis. Next, they transferred the spores from each culture into tubes containing minimal media. Fungi with auxotrophic mutations don't grow on minimal media, which allowed Beadle and Tatum to identify cultures that possess mutation. Once they determined that a particular culture had an auxotrophic mutation, they set out to determine the specific effect of that mutation. They transferred spores of each mutant strain from complete medium to a series of tubes each of which had minimal medium and one of the essential biological molecules, such as an amino acid. If the spores in a tube grew, Beadle and Tatum were able to identify the additional substance as the biological molecule whose synthesis had been affected by the mutation. For example, an auxotroph that would grow only on minimal media to which arginine had been added must have possessed a mutation that disrupts the arginine synthesis. Adrian Zerb and Horowitz patiently applied this procedure to genetically dissect the multi-step biochemical pathway of arginine synthesis, as shown here. They first isolated a series of auxotrophic mutants whose growth required arginine. Then they tested these mutants for their ability to grow on minimal medium, supplemented with 3 compounds, ornithine, citrulline, and arginine. From the results, they were able to place the mutants into 3 groups on the basis of which substances allowed growth. Based on these results, these scientists proposed that the biochemical pathway leading to the amino acid arginine has at least 3 steps, as shown over here in pathway 1. They concluded that the mutation in group 1 affects step 1 of the pathway, mutations in group 2 affect step 2, and mutations in group 3 is going to impact step 3. But how did they know that the order of the compounds in the biochemical pathway was correct? Notice that if step 1 is blocked by a mutation, then the addition of either ornithine or citrulline allows growth because these compounds can still be converted into arginine. In addition, if step 2 is blocked, the addition of citrulline allows growth, but the addition of ornithine won't have an effect. If step 3 is blocked, the spores will grow only if arginine is added to the media. The underlying principle is that an auxotroph can't make any compound that comes after the step that's blocked by a mutation. Using this reasoning with the information shown here in table 15.1, right up here in the corner, okay, we can see that the addition of arginine to the media allows 3 groups of mutants to grow. As a result, biochemical steps affected by all the mutants precede the step that results in arginine. The addition of citrulline allows group 1 and 2 mutants to grow, but not group 3. Thus, group 3 mutants must affect a biochemical step that takes place after the production of citrulline, but before the manufacture of arginine. The addition of ornithine allows the growth of group 1 mutants, but not group 2 or 3. Thus, mutations in group 2 and 3 affect steps that come after the production of ornithine. We've already established that group 2 mutations affect a step before the production of citrulline, so group 2 mutants must block the conversion of ornithine into citrulline. And that's shown here in pathway 3. Okay? Because group 1 mutations affect some steps before the production of ornithine, we can conclude that they must affect the conversion of some precursor into ornithine. We can now outline the biochemical pathway yielding ornithine, citrulline, and arginine, shown here as pathway 4. Okay? It's important to note that this procedure doesn't necessarily detect all steps in a pathway. Rather, it detects only the steps that produce the compounds that are tested. Using mutations in this type of reasoning, Beadle and Tatum and others were able to ID genes that control several biosynthetic pathways in Neurospora. They established that each step in a pathway is controlled by a different enzyme, as shown here in 15.3, right, which is this figure right here. Okay. In addition, by conducting genetic crosses and mapping experiments, they were able to demonstrate that mutations affecting any one step in a pathway always occurred at the same chromosomal location. These scientists then reasoned that mutations affecting a particular biochemical step occurred at one locus that encoded a particular enzyme. This idea became known as the one gene, one enzyme hypothesis. Genes function by encoding enzymes, and each gene encodes a separate enzyme. Although Beadle and Tatum examined encoded enzymes, a lot of genes encode proteins that are not enzymes. So more generally, their idea was that each gene encodes a single protein. When research findings showed that some proteins are composed of more than one polypeptide chain, and that different polypeptide chains are encoded by separate genes, this model was modified to become the one gene, one polypeptide hypothesis. Proteins are central to all living processes. Many proteins are enzymes, the biological catalysts that drive chemical reactions in the cell, and others are structural proteins, providing scaffolding and support for the membrane and the cell microstructure. Filaments, bone, and hair are also composed of structural protein to a large extent. Some proteins help transport substances, and others have a regulatory, communicative, or defensive function. All proteins are polymers composed of amino acids linked end to end. 20 common amino acids are found in proteins, and these are shown here, with both their 3-letter and their 1-letter abbreviations. All of the common amino acids are similar in structure. Each consists of a central carbon atom bound to an amino group, a hydrogen atom, a carboxyl group, and an R group that differs from each amino acid. Okay? So what you're looking at here are the building blocks of all proteins, right? You've got a nonpolar class, you've got a polar uncharged class, you've got an aromatic class which has in it a nonpolar and a couple of polar members, okay? You got positively charged amino acids and negatively charged amino acids, okay? So what happens when you assemble a protein is that you put the thing together using a ribosome to form peptide bonds between each of these building blocks, and then what determines the final shape of the protein, as you probably remember from GenBio or from A&P, is the fact that the nonpolar groups will seek out the other nonpolar groups, the polar groups will seek out each other, will seek out water, the negatively charged groups will seek out the positively charged groups, and the result will be what we call a native conformation, which contains 4 levels of structure, right? Primary, secondary, tertiary, and sometimes quaternary. And we'll go through each of these, okay? But the native shape is the shape in which the protein can function normally. And anything that destroys that native shape or doesn't let the protein be built to completion or substitutes components in the building blocks of the protein are going to affect its function, and often with negative consequences. A non-adaptive mutation, for instance, would produce characteristics in the organism that would make it less likely to survive long enough to breed, while an adaptive one would make it more likely to survive long enough to breed. And as a result of that, those conditions that are selected for, those proteins that provide a selective advantage, that genetic information is more likely to be passed to the offspring than the other genetic information. Okay, and over time, the accumulation of these changes can result in speciation. Okay, and so that's the idea behind evolution and natural selection. So let's take a look at protein structure.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Next thing we want to address is how we were able to figure out what the genetic code is, and we want to talk a little bit about the anatomy of the coding region and the universality of the code. Once it had been firmly established that the genetic code consists of codons that are 3 nucleotides in length, the next step was to determine which group of 3 nucleotides specify which amino acids. Logically, the easiest way to break the code would have been to determine the base sequence of a piece of RNA, add it to the test tube containing all the components needed for translation, and let direct synthesis occur. The amino acid sequence of the newly made protein could then be determined, and its sequence could be compared to the RNA. However, there was no way at the time to determine the nucleotide sequence of a piece of RNA, so indirect methods were used. The first clues to the genetic code came in '61 with work from Nirenberg and Mathai. These investigators made synthetic RNAs using an enzyme called polynucleotide phosphorylase. Unlike RNA polymerase, polynucleotide phosphorylase does not require a template. It randomly links together any RNA nucleotides that happen to be available. The first synthetic messenger RNAs used by Nirenberg and Mathai were homopolymers, meaning they were made up of the same nucleotide linked together many times in a row. Okay? As an example, by adding a polynucleotide phosphorylase to a solution of uracil, they generated RNA molecules that consisted entirely of uracil and thus had triple U codons. These poly-U RNAs were then added to 20 test tubes, each containing the components needed for translation and all 20 amino acids. A different amino acid was radioactively labeled in each of the 20 tubes. Radioactive protein appeared in only one of the tubes, the one containing the labeled phenylalanine. And this showed that the codon U specifies the amino acid phenylalanine. The results of similar experiments using other homopolymers allowed us to determine which homopolymer triplets, were responsible for a particular amino acid being incorporated into a protein. To gain information about other codons, these scientists made synthetic RNAs containing 2 or 3 different bases. Because polynucleotide phosphorylase incorporates nucleotides randomly, these RNAs contain random mixes of bases and are thus called random copolymers. For example, when adenine and cytosine nucleotides were mixed with polynucleotide phosphorylase, the RNA molecules produced 8 different codons: AAA, AAC, ACC, ACA, CAA, CCA, CAC, and CCC. These poly-AC RNAs produce proteins containing 6 different amino acids: asparagine, glutamine, histidine, lysine, proline, and threonine. The proportions of the different amino acids in the proteins produced depended on the ratio of the 2 nucleotides that were used in creating the copolymer, and the theoretical probability of finding a particular codon could be calculated from the ratios of the bases. If a 4-to-1 ratio of C to A were used in making the RNA, the probability of C being on any given position in a codon would be 4/5, and the probability of it being of an A being in it would be 1 in 5. With random incorporation of bases, the probability of any one of the codons with 2 C's and 1 A would be 4 out of 5 times 4 out of 5 times 1 out of 5, which is essentially 13%. The probability of any codon with 2 A's and 1 C would be about 3%. Thus, an amino acid encoded by 2 C's and 1 A should be more common than one encoded by 2 A's and 1 C. By comparing the percentages of amino acids in proteins made by the random copolymers with the theoretical frequency expected by the codons, these individuals could derive information about the base composition of the codons. These experiments revealed nothing about the codon base sequence. Histidine was clearly encoded by a codon with 2 C's and 1 A, but whether the codon was ACC, CAC, or CCA was unknown. There were other problems with this method. The theoretical calculations depended on the random incorporation of bases, which didn't always occur. In addition, because the genetic code is redundant, sometimes several different codons specify the same amino acid. To overcome the limits of the random copolymers, Nirenberg and later developed another technique in '64 that used ribosome-bound transfer RNAs. They found that a very short sequence of messenger RNA, even one made of one codon, could bind to a ribosome. The codon on the short message then base pair with a matching anticodon on a transfer RNA carried an amino acid specified by that codon. When the short messages were bound to ribosomes that were mixed with transfer RNAs and amino acids, this mix was passed through a nitrocellulose filter. The transfer RNAs that were paired with the ribosome-bound message stuck to the filter, whereas unbound transfer RNAs passed through it. The advantage of this system was that it could be used with very short synthetic messenger RNA molecules that could be made with a known sequence. Nirenberg and later made over 50 short messages with known codons and then individually added them to a mix of ribosomes and transfer RNAs with amino acids. They then isolated the transfer RNA that was bound to the message and ribosomes and determined which amino acids were present on the bound RNAs. For example, synthetic messenger RNA with the codon GUU retained a transfer RNA with valine, whereas messenger RNAs with UGU and UUG didn't. Using this method, Nirenberg and his colleagues were able to determine the amino acids encoded by over 50 codons. So let's look at the triplet code and discuss what a sense codon and a stop codon are. Okay? We are going to note that this is a degenerate genetic code, meaning that more than one triplet can specify a particular amino acids. Iso-accepting transfer RNAs are different transfer RNAs that accept the same amino acid but have different anticodons. One amino acid is encoded by 3 consecutive nucleotides in a messenger RNA, and each nucleotide can have one of 4 possible bases, being A, G, C, or U. So there are 43, which equals 64 possible codons. 3 of these codons are stop codons, which specify the end of translation, as we'll see shortly. Thus, 61 codons called sense codons encode the amino acids. Because there are 61 sense codons and only 20 different amino acids commonly found in any given protein, the code contains more information than is needed to specify the amino acids in the protein, and thus is said to be a degenerate code. This expression doesn't mean that the genetic code is depraved. Degenerate is simply a term that Crick borrowed from quantum physics, where it describes multiple physical states that have equivalent meanings. The degeneracy of the genetic code means that the code is redundant. Amino acids may be specified by more than one codon. Only tryptophan and methionine are encoded by 1 codon. Other amino acids are specified by 2 or more, and some, such as leucine, are specified by 6 codons. Codons that specify the same amino acid are said to be synonymous, just as synonymous words are different words that have the same meaning. Okay? So this is why, all right, you can sometimes have a mutation in the coding region of DNA that shows up in the messenger RNA that does not change the amino acid sequence of the protein. Okay? And then the next thing we want to address is why is it that when we look at the degeneracy of the code that it seems to be the first 2 nucleotides are more critical than the third one. And that has to do with something called the wobble hypothesis, which we'll get to now, right? As we learned in an earlier chapter, the last chapter, transfer RNAs are the adapter molecules that bind particular amino acids and deliver them to the ribosome, where the amino acids are then assembled into the polypeptide chain. Each type of transfer RNA attaches to one type of amino acid. The cells of most organisms have about 30 to 50 different transfer RNAs, yet there are only 20 different amino acids commonly found in the proteins. Thus, some amino acids are carried by more than one transfer RNA. Different transfer RNAs that accept the same amino acid but have different anticodons are called isoaccepting transfers. Even though some amino acids can pair with multiple transfer RNAs, there are still more codons than anticodons. One anticodon can pair with different codons through flexibility in base pairing at the third position of the codon. Examination of the structure of the transfer RNA shows that many synonymous codons differ in only the third position. We saw that in the table earlier as well. For example, serine is encoded by the codons UCU, UCC, UCA, and UCG, all of which begin with UC. When the codon of the message and the anticodon of the transfer RNA join, the first base of the codon forms a hydrogen bond with the third base of the anticodon, strictly according to Watson and Crick base pairing rules, A with U, C with G. Next, the middle bases of the codon and anticodon pair, so strictly following the Watson and Crick rules for the first 2 positions, and after these pairs have bonded, the third base pair is going to weakly bond, and as a result there's some flexibility or wobble in their pairing, and that's the whole idea behind the wobble hypothesis. In '66, Crick developed the wobble hypothesis, which proposed that some non-standard pairings of bases could take place at the third position of a codon. For example, a G in the anticodon could pair with either a C or a U in the third position of that codon. The important thing to remember about wobble is that it allows some transfer RNAs to pair with more than one message codon. Thus, from 30 to 50 transfer RNAs can pair with 61 sense codons. And some codons are synonymous as a result of this wobble. Usually, however, each nucleotide is part of one codon. A few overlapping genes are found in viruses, but codons within the same gene don't overlap, and the genetic code is generally considered to be non-overlapping. For any sequence of nucleotides, there are 3 potential sets of codons, 3 ways in which the sequence can be read in groups of 3 each, with different ways of reading the sequence, and these are called reading frames. And any sequence of nucleotides that has 3 potential reading frames represents a problem, right? how does the reading frame begin? How does it end? Okay, so how does the ribosome determine which AUG is the real deal? Okay, and that's what the Kozak sequence is all about. That's this bind and scan hypothesis where the ribosome latches on to proteins that are associated with the cap and then starts to wander down the 5' untranslated, and it'll encounter several AUGs, but it's only the AUG that's in the proper sequence context that's going to let that ribosome pause at that point and begin initiation of translation. Okay, so that's the key. All right. So it's essential for the translation machinery to use the correct reading frame, and how do we establish that? The reading frame is set by the initiation codon, which is the first codon of the message to specify an amino acid. After the initiation, the other codons are read as successive groups of 3 nucleotides. No bases are skipped between the codons, so there's no punctuation marks to separate them. The initiation codon is an AUG, although sometimes GUG and UUG are used on rare occasion. The initiator is not just a sequence that marks the beginning of translation, it also specifies an amino acid. In bacterial cells, the first AUG encodes a modified type of methionine called N-formyl methionine, or N-form-met for short. As a result, all proteins in bacteria begin with this amino acid, but its formal group can be removed after the protein's been produced. When the codon AUG is at an internal position in a gene, it encodes unformulated methionine. In archaea and eukaryotic cells, AUG specifies unformulated methionine, both at the initiation position and at the internal positions. In bacteria and eukaryotes, there are different transfer RNAs for the initiator methionine and internal methionines. Okay? 3 codons, UAA, UAG, and UGA, don't encode amino acids. These codons, which signal the end of translation in both bacterial and eukaryotic cells, are stop codons or termination codons, or sometimes they're called nonsense codons. No transfer RNAs have anticodons that pair with termination codons, unless there's been a mutation in a transfer RNA gene. Okay? For many years, the genetic code was assumed to be universal, meaning that each codon specifies the same amino acid in every organism. We now know that the genetic code is mostly, but not completely, universal. Some exceptions have been found. Most of these are termination codons, but there are a few cases in which one sense codon substitutes for a different one. Many of these exceptions are found in mitochondrial genes. Some non-universal codons have also been detected in the nuclear genes of protozoa and in bacterial DNA, and the table here shows some of those. One study of bacteria and bacteriophage isolated 1,776 environmental samples found in non-universal codons in a substantial fraction, suggesting that non-universal codons may be more common than we once thought. Now that we're familiar with the genetic code, we can start to talk about how the amino acids are assembled into protein. Because more is known about translation in bacteria than in eukaryotes, we'll focus on bacterial translation. In most respects, eukaryotic translation is similar, although some significant differences are going to be pointed out. Remember that only messenger RNAs are transferred into protein. Translation takes place on the ribosome. In fact, the ribosome is the protein synthesizing factory. Through many techniques, a detailed view of the structure of the ribosome has been produced. In recent years, this has greatly improved our understanding of translation. A ribosome attaches near the 5' end of the message strand and moves toward the 3' end, translating the codons as it goes. Synthesis begins at the amino end of the protein and ends at the carboxyl end, with elongation taking us from the amino to the carboxyl terminus. Protein synthesis includes a series of RNA-RNA interactions between the message and the ribosomal RNA that hold the messenger RNA in the ribosome. Between the codon on the messenger RNA is the anticodon on the transfer RNA, and between the transfer RNA and the ribosomal RNA is the ribosome. Okay? So the ribosome grips the transfer RNA everywhere but at the anticodon, and the message grips the transfer RNA at the anticodon, and the message itself is also gripped by the ribosome. Protein synthesis can be divided into 4 stages. tRNA charging, in which the transfer RNA binds to the amino acids. Initiation, in which the components needed for translation are assembled at the ribosome, an elongation in which amino acids are joined, one at a time, to the growing polypeptide chain, and finally termination, in which protein synthesis stops at the termination codon and the translation components are released from the ribosome. The first stage of translation is the binding of the transfer RNA molecules to the correct amino acid. As we've seen, each transfer RNA is specific for a particular amino acid, and all transfer RNAs have the sequence CCA at their 3' end, and the carboxyl group of this amino acid is attached to the adenine nucleotide at the 3' end of the transfer RNA. If each transfer RNA is specific for a particular amino acid, but all amino acids are attached to the same nucleotide at the 3' end of the transfer RNA, how does the transfer RNA link up with the correct amino acid? So that's a question we'll answer now. The key to specificity between an amino acid and its transfer RNA is a set of enzymes called aminoacyl tRNA synthetases. The cell has 20 different synthetases, one for each of the 20 different amino acids. Each synthetase recognizes a particular amino acid as well as all the transfer RNAs that accept that amino acid. Its recognition of the correct amino acid is based on the different sizes, charges, and R groups of the amino acids. Its recognition of the correct transfer RNA depends on the nucleotide sequence of the transfer RNA. Scientists have identified which nucleotides are important in recognition by synthetases by altering different nucleotides in a particular transfer RNA and determining whether the altered transfer RNA is still recognized by the synthetase. The attachment of a transfer RNA to its correct amino acid, termed tRNA charging, requires energy which is supplied by ATP. So the chemical reaction is shown here, right? Amino acid and transfer RNA and ATP gives us aminoacyl tRNA, AMP, and pyrophosphate. And again, it's cleaving that phosphate bond that generates the energy captured to drive the process. Otherwise, it wouldn't happen spontaneously. This reaction takes place in 2 steps. To ID the resulting amino-acylated tRNA, we write the 3-letter abbreviation for the amino acid in front of the transfer RNA. As an example, the amino acid alanine attaches to its transfer RNA, producing its aminoacyl tRNA. Errors in tRNA charging are rare. They occur in only about 1 in 10,000 to 1 in 100,000 reactions. This is due in part to the presence of editing activity on many of the aminoacyl tRNA synthetases. Editing activity detects incorrectly paired amino acid and removes them from the transfer RNA. Some antifungal chemical agents work by trapping tRNAs in the editing site of the synthetase, preventing their release and inhibiting the process of translation in the fungus, and that's the basis of the fungicide. Okay? The second step in the process of protein synthesis is initiation. At this stage, all the components needed for protein synthesis assemble. The first is the message, then the small and large subunits of the ribosome, and then a set of 3 proteins called initiation factors, and finally, initiator tRNA with N-formyl methionine attached, and we abbreviate that FMET tRNA IF-MET, and 5-guanosine triphosphate, which is going to be the energy source for this reaction, just like ATP is for many other reactions. The initiation comprises 3 major steps. First, the message binds to the small subunit of the ribosome. Second, the initiator transfer RNA binds the messenger RNA through base pairing between the initiation codon and the anticodon. And finally, the large ribosomal subunit joins the initiation complex. So now let's look at these steps a little bit more closely. The functional ribosome of bacteria exists as 2 subunits, the small 30S and large 50S subunit. A messenger RNA molecule can bind to the small ribosomal subunit only when they're separated. Initiation factor 3, also known as IF3, binds the small ribosomal subunit and prevents the large subunit from binding during initiation. Another factor, initiation factor 1, enhances the dissociation of the large and small subunits. Where on the messenger RNA do the ribosomes bind during the initiation of translation? Key sequences on the message are required for the ribosome binding, which have been identified by techniques designed to allow a ribosome to bind to message but not proceed with protein synthesis. The ribosome is stalled at this point, and ribonuclease is added, but the ribonuclease can't digest the area of the message that's under the ribosome. So this is an example of something called RNase protection assay. And then we can recover the protected RNA sequence, and we can generate a cDNA from it using reverse transcriptase, and we can sequence it. Okay. The sequence covered by the ribosome during the initiation is 30 to 40 nukes along the region bracketing the AUG, and it includes what are called the Kozak sequences. The initiator transfer RNA attaches to the initiation codon. This attachment requires the initiation factor 2 and forms a complex with GTP, which is the energy. At this point, the initiation complex has the small ribosomal subunit, the message, the initiator transfer RNA and its amino acid, a molecule of GTP, and several initiation factors. These components are collectively known as the 30S initiation complex. In the final step of initiation, IF3 dissociates from the small subunit, allowing the large ribosomal subunit to join the initiation complex. The molecule of GTP is hydrolyzed to guanosine diphosphate, and the initiation factors dissociate from the complex. When the large subunit has joined the initiation complex, it's called the 70S initiation complex. Now you might well ask, okay, why don't these S numbers add up in the ribosome? And the answer is that the Svedberg unit is a measure of density, which is both a mass per unit volume, but also a measure of the type of volume that the particle has. Okay, it is not strictly a mass measurement. So the assembled ribosome can have a different shape than the individual subunits will have, and will sediment at a different area in a sedimentation gradient, in a centrifuge. Okay. When the ribosome binding site is within the ribosome binding site is the Shine-Dalgarno sequence for bacteria, and it's the Kozak sequence for eukaryotes. Okay, this is a consensus sequence complementary to the sequence of the nucleotides at the 3' end of the 16S ribosomal RNA. During initiation, the nucleotides in Shine-Dalgarno pair with their complementary nucleotides in the 16S ribosomal RNA, and this allows the small ribosomal subunit to attach to the message and position the ribosome over the initiation codon. These ribosome binding sequences are within the 5' untranslated region of the message. Similar events take place in the initiation of translation to eukaryotes. Okay, so what we're talking about here is an analogous process that uses slightly different components but accomplishes the same thing. Okay. Some important differences in bacterial cells, sequences in the 16S RNA of the small ribosomal unit bind the Shine-Dalgarno in the message. No analogous consensus exists in eukaryotic message RNA. Instead, the cap at the 5' end of the eukaryotic message is critical in the initiation of translation. In a series of steps, the small subunit of the eukaryotic ribosome, initiation factors, and the initiator transfer RNA with its amino acid form a complex that recognizes the cap and binds there. The initiation complex then moves along or scans the 5' UTR in the message until it locates the first AUG. This is called the bind and scan model. Okay, the identification of the start codon is aided by the Kozak sequences that surrounds it. Okay, so that's where and how the ribosome in the eukaryote knows to pause. Okay, because it'll hit several AUGs along the way. Another important difference is that eukaryotic initiation has 12 initiation factors. Some of these factors keep the ribosomal subunit separated, just as IF3 does in bacteria. Others recognize the 5' cap in the message and allow the small ribosomal subunit to bind there. Still others have RNA helicase activity used to unwind secondary structure that can exist in the 5' untranslated region of the message, allowing the small subunit to move down the messenger RNA until the initiation codon is reached. Other initiation factors help bring the initiation complex to its location on the message prior to the beginning of translation. In eukaryotes, the 5' cap is bound by several proteins, one of which is the cap binding complex, or CBC, which aids in exporting the message from the nucleus and then promotes the pioneer or initial round of translation in the cytoplasm. The first round of translation plays an important role in checking for errors in the message. After the pioneer round of translation, the CBC is replaced by the eukaryotic initiation factor 4E, which promotes continued translation of the message. The poly A tail at the 3' end of the eukaryotic message also plays a role in the initiation of translation. During initiation, proteins attached to the poly A tail interact with proteins that bind to the 5' cap, enhancing the binding of the small ribosomal subunit to the 5' end of the message. This interaction indicates that the 3' end of the message bends over and associates with the 5' cap during the initiation of translation, forming a circular sequence known as a closed loop. A few eukaryotic messages contain internal ribosome entry sites, or irises, where ribosomes can bind directly without first attaching to the 5' cap. Furthermore, some uncapped messages are translated through the binding of initiation factors and ribosomes to modified adenine nucleotides in the messenger RNA. Okay? What's interesting about these irises is that we can use them to make artificial constructs that can basically be translated to form more than one protein product without requiring alternative splicing. Okay? Kind of interesting. The next stage in protein synthesis is elongation, in which amino acids are joined to create a polypeptide chain. This requires a 70S initiation complex that we talked about earlier, transfer RNAs with their appropriate amino acids loaded, several elongation factors, and GTP for power. A ribosome has 3 sites that can be occupied by transfer RNAs: the aminoacyl site, the peptidyl site, and the exit site. The initiation of transfer RNA immediately occupies the P site, which is the only site to which the initiation complex will bind, but all the other transfer RNAs first enter the A site. At the end of the initiation, the ribosome is attached to the message, and the FMET tRNA IF-MET complex is positioned over the AUG start in the P site. The adjacent A site is unoccupied. Elongation is a 3-step process. The first step has a charged transfer RNA bind to the A site. This binding takes place when elongation factor EF2, spelled cap E, cap F, dash cap T, small u, right, joins with GTP and then a charged transfer RNA to form a 3-part complex. This complex then enters the A site of the ribosome, where the anticodon on the transfer RNA pairs with the codon on the message. Once the charged transfer RNA is in the A site, GTP is cleaved to form GDP, and the EF2 GDP complex gets released. Elongation factor TS, which is also sometimes abbreviated cap E, cap F, dash cap TS, regenerates EF2 GDP to EF2 GTP. In eukaryotic cells, a similar set of reactions delivers a charged transfer RNA to the A site. The second step of elongation is the formation of a peptide bond between the amino acids that are attached to the transfer RNAs in the P and the A sites. The formation of this bond releases the amino acid in the P site from its transfer RNA. Peptide bond formation occurs within the large ribosomal subunit. Catalytic activity that creates the peptide bond is a property of one of the ribosomal RNA components of the large subunit, which will be the 23S ribosomal RNA in bacteria and the 28S in eukaryotes. This ribosomal RNA acts as a ribozyme. The third step is the elongation portion, known as translocation. The movement of the ribosome down the messenger RNA is in the 5' to 3' direction. This step positions the ribosome over the next codon and requires elongation factor G, EFG, and the hydrolysis of GTP to GDP. Because the transfer RNA in the P and the A sites are still attached to the message by the codon-anticodon pairing, they don't move with the ribosome as it translocates. As a result, the ribosome shifts so that the transfer RNA that previously occupied the P site now is in the E site, from which it can then move into the cytoplasm, where it can be recharged with another amino acid. Translocation also causes the transfer RNA that occupied the A site to occupy the P site, leaving the A site open. Thus, the progress of each transfer RNA through the ribosome in the course of elongation can be summarized as cytoplasm, A site, P site, E site, then back to cytoplasm. As stated earlier, the initiator transfer RNA is the exception. It attaches directly to the P site and never occupies the A site. After translocation, the A site of the ribosome is empty and ready to receive the transfer RNA specified by the next codon. The elongation cycle repeats itself. A charged transfer RNA and its amino acid occupy the A site. A peptide bond is formed between the amino acids in the A and the P sites, and the ribosome translocates to the next codon. Throughout the cycle, the polypeptide chain remains attached to the transfer RNA in the P site. Another protein called elongation factor P, or EFP, enhances the translation of proteins that contain consecutive copies of the amino acid proline. If EFP is absent, ribosomes often stall during the translation of these polyproline-containing proteins, probably because of their shape. Okay, proline produces bends in the polypeptide chain. mRNAs, although single-stranded, often contain secondary structures formed during the pairing of complementary bases on different parts of the messenger RNA. As the ribosome moves along the message, these secondary structures are unwound by helicase activity in the small ribosomal subunit. Protein synthesis ends when the ribosome translocates to the termination codon, where there are no transfer RNA anticodons complementary, so no transfer RNA enters the A site of the ribosome. Instead, proteins called release factors bind to the ribosome. E. coli has 3 release factors, RF1, 2, and 3. RF1 binds to the termination codons, UAA and UAG, while RF2 binds to UGA and UAA. The binding of RF1 or RF2 to the A site of the ribosome promotes cleavage of the transfer RNA in the P site from the polypeptide chain and the release of the polypeptide. Release factor 3 forms a complex with GTP and binds to the ribosome. This complex brings about conformational change in the ribosome, releases RF1 or 2 from the A site, and causes the transfer RNA in the P to move to the E site. In the process, GTP is hydrolyzed to GDP. Additional factors bring about the release of transfer RNA from the P site, the release of the message from the ribosome, and the dissociation of the ribosome. It's important to note that the termination codon isn't located at the 3' end of the message. Rather, the termination codon is followed by a number of nucleotides that constitute the 3' untranslated region of the message. This region often contains sequences that affect stability of the message and influence whether translation happens. Recent study has shown that some bacterial ribosomes engage in a type of proofreading similar to the proofreading that DNA polymerases perform during replication. After translocation, the ribosome checks the interaction between the message and the transfer RNA in the P site. If the wrong transfer RNA was added, the alignment between the message and transfer RNA will be incorrect, and this triggers premature termination of translation. Evidence shows that an important function of RF3 is to help bring about termination of translation when the wrong transfer RNA is used. Translation in eukaryotes terminates in a similar way, except there are 2 release factors, ERF1, which recognizes all 3 terminator codons, and ERF2, which binds GTP and stimulates the release of the polypeptide chain from the ribosome. No translational proofreading, such as that in bacteria, has been observed in the eukaryote. So let's look at bacterial translation.
In translation, amino acids are assembled into proteins. Bacterial translation is initiated in 3 steps. First, the small subunit of the ribosome binds to the mRNA so that the P site is positioned over the start codon. Second, the initiator tRNA with N-formylmethionine attached binds to the mRNA through base pairing between the codon and the anticodon. Third, the large subunit of the ribosome joins the complex to complete initiation. During elongation, amino acids are joined to a growing polypeptide chain. The first step of elongation is the delivery of tRNA and its amino acid to the A site. In the second step of elongation, a peptide bond forms between the amino acids. The third step of elongation is translocation, the movement of the ribosome down the mRNA. After translocation, the tRNA formerly in the P site is now in the E site, from which it moves to the cytoplasm. Now the elongation cycle repeats itself. A tRNA and its amino acid occupy the A site. A peptide bond forms and the ribosome translocates to the next codon. As the cycles of elongation continue, the polypeptide chain grows by the addition of amino acids to its carboxyl end. The last stage of translation, called termination, takes place when the ribosome reaches a termination codon. Release factors bind to the termination codon, causing the release of the polypeptide chain. The ribosome then translocates one last time. The tRNA and release factors exit the ribosome, and then the ribosome dissociates. Remember that several accessory proteins not shown here also participate in translation.
The overall process of translation is shown here. There's a nice summary slide for you. The central role of the ribosome in protein synthesis was recognized in the 50s, and a lot of aspects of its structure have been studied since. Nevertheless, a lot of aspects remained a mystery until three-dimensional reconstructions were completed. In 2009, the Nobel Prize in Chemistry was awarded to the CITES and Yonath lab for their research on the structure of the ribosome, and it's shown here. This is an E. coli ribosome at low resolution. A high-resolution image of the bacterial ribosomal structure, as determined by X-ray crystallography, is shown at the bottom of the page. A messenger RNA is bound to the small ribosomal subunit, and transfer RNAs are loaded in the A, P, and E sites, which bridge the large and small subunits as shown here. Initiation factors 1 and 3 bind to sites on the outside of the small subunit. EF2, EFG, and other factors complex with GTP interact with a factor binding center. High-resolution crystallography provides information indicating that a decoding center resides in the small subunit. This center senses the fit between the codon on the message and the anticodon of the incoming charge transfer RNA, and only transfer RNAs with the correct anticodon are bound tightly by the ribosome. These structural analyses indicate that peptide bond formation occurs within the large ribosomal subunit and is carried out by the 23S RNA molecule. These analyses also reveal that a tunnel connects the site of peptide bond formation with the back of the ribosome. The growing polypeptide chain passes through the tunnel to the outside of the ribosome. The tunnel can accommodate about 35 amino acids of the growing chain as it moves down the message. In both prokaryotic and eukaryotic cells, message molecules are translated simultaneously by multiple ribosomes. It's called the polysome. The resulting structure, the polysome, has each ribosome successively attached to the ribosome binding site at the 5' end of the message and moving towards the 3' end. The polypeptide associated with each ribosome becomes progressively longer as the ribosome moves along the message. In prokaryotes, transcription and translation are simultaneous. They're in the cytoplasm. Multiple ribosomes can be attached to the 5' end of the message, while transcription is still happening at the 3' end. As shown here, in eukaryotes, transcription and translation are separated in time and space. Transcription in the nucleus and translation in the cytoplasm. The accurate transfer of genetic information from one generation to the next and from genotype to phenotype is important for the proper development and functioning of an organism. Thus, cells have evolved a number of quality control mechanisms to ensure the accuracy of the information transfer. Protein synthesis is no exception. Several mechanisms, collectively termed messenger RNA surveillance, exist to detect and deal with errors in message that can create problems in the course of translation. These mechanisms keep the cell from wasting resources in translating messenger RNAs that are improperly formed and prevent the production of truncated proteins, which are toxic to the cell. A common type of mutation is the alteration of a codon that specifies an amino acid to become a termination codon. This is called a nonsense mutation. This mutation doesn't affect transcription, but translation ends prematurely when the termination codon is encountered. The resulting protein is truncated and often non-functional. Nonsense mutations often come in eukaryotic transcription when one or more exons are skipped or improperly spliced. Improper splicing leads to the deletion or addition of nucleotides in the message. This alters the reading frame and introduces premature terminating codons. To prevent the synthesis of aberrant proteins resulting from nonsense mutations, eukaryotes have evolved a mechanism called nonsense-mediated messenger RNA decay, which results in the rapid elimination of messages containing premature termination codons. The mechanism responsible for NMD is not completely understood. In mammals, it appears to entail proteins that bind to exon-exon junctions. These exon junction proteins may interact with enzymes that degrade the message. One possibility is that the first ribosome to translate a message, which is the pioneer, removes the exon junction proteins, thus protecting the message from degradation. However, when the ribosome encounters a premature termination codon, it doesn't cross the entire message, and some of the exon junction proteins are not removed, and that results in the breakdown of that message. Nonsense-mediated message decay does not occur in bacteria. A problem that sometimes comes in translation is the stalling of the ribosome on the message before translation ends. This can happen because a mutation in DNA can change a termination codon into one that specifies an amino acid. It can also come when transcription terminates prematurely, producing a truncated message lacking a termination codon. In these cases, the ribosome reaches the end of the message without encountering a terminator, whereupon it stalls, attached to the message. Attachment to the message prevents the ribosome from being recycled for use on other messages. If such an occurrence is frequent, the result is a shortage of ribosome that diminishes the overall level of protein synthesis. Basically, they all get backed up on the message. They get plugged in there. Bacteria have evolved a molecular tow truck called transfer messenger RNA, called tmRNA for short, that removes stalled ribosomes. This RNA molecule has the properties of both a tRNA and an mRNA. Its tRNA component is normally charged with the amino acid alanine. When a ribosome becomes stalled on a message, EF2 delivers the tmRNA to the ribosome A site, where it acts as a surrogate tRNA. A peptide bond made between the amino acid in the P site of the ribosome and the alanine attached to the tmRNA, now in the A site, transfers the polypeptide chain to the tmRNA, releasing the tRNA in the P site. The ribosome then resumes translation, switching from the original aberrant message to the message part of the tmRNA, adds 10 amino acids encoded by the tmRNA, and then reaches a termination codon at the 3' end of the tmRNA, which terminates translation and releases the ribosome. The additional amino acids act as a tag that targets the incomplete polypeptide chain for breakdown. Some evidence suggests that the tmRNA also targets aberrant message for degradation. How stalled ribosomes are recognized by tmRNA isn't clear, but this method is efficient at recycling stalled ribosomes and eliminating abnormal protein from truncated transcription. Eukaryotes have evolved a different mechanism to deal with messages that are missing termination codons. Instead of restarting the stalled ribosome and degrading the abnormal protein that results, eukaryotes use a mechanism called nonstop message decay, which results in the rapid breakdown of abnormal message. In this mechanism, the codon-free A site of the stalled ribosome is recognized by a specific protein, which binds to the A site and recruits other proteins, which then break down the message from the 3' end. Another message surveillance system is found in eukaryotes known as no-go decay, which helps remove stalled ribosomes, resulting from secondary structure in the message, chemical damage to the message, premature stop codons, and ribosomal defects. A series of proteins bring about termination, recycling of the ribosome, and breakdown of the messenger RNA. Okay? All right. I thank everybody for listening, and I will see you in the next podcast.